MERS-CoV, also known as the Middle East Respiratory Syndrome coronavirus, first emerged in late 2012. Since its emergence, a total of 2,279 patients from 27 countries have been infected across the globe. To date, approximately 806 patients have died, with a death rate of 40 to 60% as reported by the World Health Organization (WHO) in February 2019. MERS primarily is an airborne disease which spreads through an infectious patient's cough and sneezes. However, the exact way the virus spreads is unknown. MERS has spread from many mediums, from taking care of an infected person at home and hospitals to camels. Yes, you heard camels. In areas with high rates of MERS, the WHO recommends avoid having close contact with camels, drinking raw camel milk or urine, and eating undercooked camel meat. The main people at risk of MERS include recent travelers of the Arabian Peninsula suffering from respiratory issues within 14 days of return and individuals who are in close contact with an affected person. The risk of contracting the virus also increases with age, thus elderly with weakened immunity are easily exposed to the virus. Incubation period, aka the time taken from the viral exposure to the start of symptoms, appearing usually about 5 to 14 days. Common signs and symptoms of MERS include fever, cough, muscle pain, and vomiting. These mild symptoms can escalate to severe cases of high fever and even death. Unfortunately, to date, there is no effective approved treatment or vaccine for MERS-CoV. Thus, there is an urgent need to develop novel MERS therapies and vaccinations to mitigate the risk of potential pandemics. So how do you fight MERS? Naturally, our body has two immune responses when under attack from the MERS virus. The virus uses its spike proteins as adhesive factors that are pro-inflammatory for host entry through a specific receptor called dipeptidic peptidase 4. This receptor is considered a key factor in the signaling and activation of the acquired and innate immune responses in infected patients. Using potent antigens in combination with strong adjuvants may effectively trigger the activation of specific MERS-CoV cellular responses as well as the production of neutralizing antibodies. This can be seen when the MERS virus infects macrophages to DPP4 binding. The macrophages then present the MERS antigens to Th0 cells, which leads to T cell activation and differentiation of the production of various cytokines associated with the different T cell subsets, including Th1, Th2, and Th17. The release of IL-12 leads to the activation and differentiation of Th0 cells to Th1 cells. Th1 cells then produce IL-2 and IFN gamma. IL-2 regulates the proliferation and development of CD8 plus T cells after antigen stimulation. IL-2 is also essential for introduction of effector cytotoxic T cell differentiation and development of CD8 plus memory T cells. CD8 plus T cells are then developed and produce IFN gamma and granzyme, which helps with the clearance of MERS virus. Mechanism involves the attachment of MERS to DPP4 on the host cell through a protein, leading to the appearance of viral genomic RNA in the cytoplasm. Immune responses will then be partially generated to double-stranded RNA during viral replication. A toll-like receptor, TLR3, synthesized by double-stranded RNA and cascades of signaling pathways are activated to produce type 1 IFN and pro-inflammatory cytokines, IL-1, IL-12, and IL-18. TLR3 also helps regulate activity of the immune system and enhances release of antiviral proteins to protect uninfected cells. Virus cell interactions lead to strong production of immune mediators. Secretion of large quantities of cytokines and chemokines is promoted in infected cells in response to MERS infection. Cytokines and chemokines then recruit lymphocytes and leukocytes to the site of infection. So how does the virus evade the immune system? Accessory proteins of MERS virus can interfere with TLR3 signaling and bind to double-stranded RNA of MERS virus during replication to prevent activation of TLR3 and evade the immune system. The affected cells have no protection due to the lack of the type 1 IFN produced by the unaffected TLR3. Pro-inflammatory cytokines are also not produced by TLR3 and leukocytes and lymphocytes are unable to be recruited to the site of infection, hence the virus is able to evade the immune system. So how does the virus replicate? Coronavirus has spike proteins that are activated by protease, which helps mediate membrane fusion and entry into the host cells. The location of the spike protein where protease activates the process is known as the cleavage site. Upon entering a cell, viral contents are released. Viral RNA is then released and enters the nucleus of the cell where it is replicated by RNA polymerase to form viral RNA. Viral RNA is then translated into viral proteins. The viral proteins are then packaged to form new viruses which are then released into the cell. The new virus then finds a new host cell to repeat the replication process. We will now be talking about the regulation of type 1 and type 2 major histocompatibility complex (MHC for short) genes that lead to the interaction between antigen-presenting cell (APCs) and T cells. Effector macrophages present viral antigens using MHC class 1 coupled with MHC class 2 to help T and B cells elicit the cellular and humoral immune responses. For MHC class 1s, proteins are fragmented into cytosol by proteasomes or other proteases. Specialized molecules known as TAP transport the peptide from the cytosol into the ER. Synthesis and assembly of class 1 heavy chain, B2 microglobulin, and the peptide form a stable complex that is transported to the cell surface. For MHC class 2, protein antigens are ingested by the APCs. Antigen processing then takes place. The MHC produced by ER with class 2 associated invariant chain peptide clip for short is formed. The clip is then removed to expose antigenic binding site for peptide binding, and the peptide MHC complex is then presented at the cell surface. Through this video, we have talked about both the innate and adaptive immune responses to MERS-CoV and the current state of MERS-CoV vaccine development. We also hope that this study has helped increase your understanding of the mechanism that enhance the MERS-CoV immune response and subsequently contribute to control of MERS-CoV infections. Thank you very much. Hope you enjoyed the video.